¿Qué tal? Buenas tardes. Continuamos con nuestra sesión de conferencias plenarias en este 8º Congreso Internacional de Matemáticas y sus aplicaciones. Nuestro siguiente conferencista es Vitaly Volpert, un investigador senior del Centro Nacional de Investigaciones Científicas, CNRS, en Francia, y director del Centro de Investigación Interdisciplinar Modelización Matemática en Biomedicina de la Universidad RUDN en Rusia. Obtuvo su doctorado en el Instituto de Física Química de la Academia de Ciencias de la URSS y habilitación en la Universidad de Lyon. Sus intereses científicos incluyen ecuaciones diferenciales parciales y sus aplicaciones a la modelización matemática. Es autor de unas 400 publicaciones científicas, incluidas 4 monografías. Es fundador y editor en jefe de la revista Mathematical Modeling of Natural Phenomena. Our next plenary speaker is Vitaly Volpert. Vitaly Volpert is a senior researcher at the National Center for Scientific Research, CNRS, France, and director of the Interdisciplinary Research Center, Mathematical Modeling in Biomedicine, at the RUDN University in Russia. He got his PhD at the Institute of Chemical Physics of the USSR Academy of Sciences and habilitation at the University of Lyon. His scientific interests include partial differential equations and their applications to mathematical modeling. He is an author of about 400 scientific publications, including 4 monographs. He is a founder and editor-in-chief of the journal Mathematical Modeling of Natural Phenomena. Welcome, Professor Volpert. He will tell us about reaction-diffusion equations in biological and biomedical applications. Welcome, is your time. Please.
Thank you very much for this presentation, and thank you all also for the invitation to participate in this conference. And as it is announced, the title of my today's lecture is Reaction-diffusion Equations in Biomedical Applications. And I will begin with a short introduction to the theory of reaction-diffusion equations. I will show some applications where such equations arise. Then we will shortly discuss some mathematical questions related to existence of certain solutions of these equations, namely of waves and pulses. And in the second part of my presentation, I will discuss some biomedical applications of reaction-diffusion equations. I will talk a little bit about cardiovascular system and diseases, and about viral infection and immune response. So here you see in the slide this classical form of the reaction-diffusion equation. The function u is a function of 2 variables, x, its space variable, and t and time. It describes the distribution of temperature or concentration, or the density of some population in population dynamics, for example. And the first term on the right side, in the case of mass concentration, it's a classical diffusion term, or it can be heat diffusion, or for example, it can be a random motion of some individuals in the case of population dynamics. And the second term, sometimes called the source term, describes heat or mass production or reproduction rate or something similar. So we can consider just one equation as it is shown here in the slide, or we consider a system of such. The theory of reaction-diffusion equations begins already almost 100 years ago in the 1930s with the applications to heat explosions. There were some first works by Semyonov and Frank Kamenetsky. Then also another very important application in combustion theory and in population dynamics by famous works by Fisher and KPP. KPP stands for Kolmogorov, Petrovsky, and Piskunov for the propagation of dominant gene, and the works by Zygdeutscher, Frank Kamenetsky in combustion theory. And the third application which determined the development of the theory was pattern formation and the famous work by Turing in 1952. So I will briefly recall some of these applications to show how this theory emerged and some of the basic notions and results. So the first application which determines the development of reaction-diffusion equations was the problem of heat explosion. And here you see the classical Frank-Kamenetsky model of heat explosion. Mathematically speaking, it is semilinear parabolic equation with this exponential term in the right-hand side, which describes heat production by chemical reaction. Of course, in spite of the fact that it is a null problem, it still remains very important. You see, for example, in the right figure in the slide, the recent explosion in Beirut, which was a terrible event, of course, and the consequences of this explosion continues till now. And from the mathematical point of view, heat explosion can be related to the existence and stability of stationary solutions of this equation. You see in the lower figure. So the temperature distribution converges to the stationary distribution, temperature remains bounded, and there is no heat explosion. However, depending on parameters of this problem and on the spatial domain, the stationary solution of this equation may not exist, and in this case, the temperature will have unbounded growth. Of course, it's a mathematical approximation. In reality, of course, it remains bounded, and this unbounded growth of temperature corresponds to heat explosion. It's a very good example which shows that mathematical properties and mathematical problems such that existence of solutions, stability of solutions, and their bifurcation have very, very clear and very important applications. The next important example related to propagation, mentioned already here, the work by Kolmogorov, Petrovsky, Piskunov, and Fisher in 1938. They considered the same reaction-diffusion equation, but in this case, on the whole axis, and the studied solutions, as it is shown in the lower figure. This solution, which has a constant profile, as shown here, decreasing function, and it propagates with a constant speed. And mathematically speaking, it is a solution shown here, u of xt equal w of x minus cc. This is called traveling wave solution. C here is the speed of propagation of this reaction diffusion wave. And this was the first works where these traveling waves were considered in parabolic equations. Another very interesting and important example is related to ecological invasion. This is, you see in this slide, this ecological problem. In the 20th century, this American mudboard was introduced in Europe in the beginning of the 20th century. You see here in this left figure, and these lines show the area where this animal just was found, and this area depends on time. You see, in the 1905 was a small area, and it grows in time, and this also corresponds to propagation of reaction-diffusion waves. Another important example is related to epidemic progression. You see here the propagation of seasonal influenza in the United States. It's this wave propagating from the west coast to the east coast, and this is something very specific for epidemic progression. Well, another example in this case, I mentioned combustion theory. You see here classical examples of flame propagation. Flames are also reaction diffusion waves. In the left figure, it's combustion agents, and in the right figure, forest fires. So these, of course, are examples described by reaction diffusion equations. And here, in this case, this is an example already from biomedicine. It's tumor growth. So it appears that tumor growth is also described by reaction diffusion equations, and not only by reaction diffusion equations, but more particularly reaction diffusion waves. And you see here the same equation at the top. Of course, it is a very simplified model, but still, which gives a good description. And in the bottom right figure, these are experimental results which show how tumor grows in time, these level lines, and these level lines, their propagation in space and the time again correspond to reaction diffusion waves. There are many other examples, and I will only mention briefly here excitable medium, propagation of neural pulses in axons and so on. In the bottom right figure, you see waves on the heart surface, also both to normal heart functioning or to heart fibrillation. And as I mentioned in the beginning, the third application which determined propagation, development of the theory is related to pattern formation. And this theory started from the Turing paper published in 1952. And here you see several examples of this. For example, pattern on the animal skin. They can be quite precisely described by reaction-diffusion equations. Is not efficient to describe this pattern formation. So in the next part of my lecture, I will very briefly discuss some mathematical properties of particular solutions, which are called waves and pulses. Of course, it will be very simple and very short, just to give an idea what it is about. So as before, we consider this reaction-diffusion equation written here in the slide, and we look for a solution shown in the top right figure. As I mentioned, this is called reaction-diffusion wave. This is a solution with a constant profile which propagates with a constant speed c here, and we can write it as uxt equal wx minus ct, c is the wave speed. And this, what is important to stress, that this wave speed is an unknown constant which should be found as a solution of this problem. If we substitute this equation, this solution wx minus ct in this equation, we will obtain the second order equation, w second derivative plus cw prime plus f of u equals 0 with some limits at infinity. And existence and properties of such solutions depend on the form of the function f of w of this form, source term or reaction term. And in applications, there are basically two main types of this function are considered. In the left lower figure, it is you see this parabolic function. It is also monostable case. Why it is like that? Because this function has two zeros here. Sorry, u equals 0 and u equal 1. These are zeros of this equation, du dt equal f of u, and u equal 1 is unstable solution, u equal 1 is stable solution. This is why this is called monostable case. And the second example, called bistable case, points 0 and 1 are stable solutions of this equation, du dt equal f of u. The third case is less interesting, and we will not discuss. Let us consider equation of wave existence in this monostable case. So we study this equation written here, w second derivative plus cw prime plus f of u equals 0. And we can write this equation as a system of two first order equations written below, w prime equal p, p prime equals minus cp minus f of w. And of course, it is very easy to study the phase plane and the trajectories on this phase plane. And in fact, what we are looking for, if we consider this equation, this system of equations, it has two stationary points written here, 1, 0, and 0, 0. And we look for a trajectory departing from the point 1, 0 and arriving to the point 0, 0. So how we can prove the existence? We will not stop in detail that. We study these stationary points, their types. It appears that the right point 1 1, 0 is a saddle point, 0, 0 is a stable node point, and we can easily show that the trajectories are shown like here. At the top they go down, at the bottom they go up. So the trajectory leaving the point 1, 0 necessarily will come to the point 0, 0. This is a very simple proof of the existence of waves, and we should remember, and this is quite simple but very important result, which is often used in the applications, that in the monostable case, such waves exist for all values of the speed greater than or equal to some minimal speed c0. Let us keep this in mind. If we consider the bistable case, the situation is a little bit different. As before, we consider this system of two first-order equations. In this case, we have two stable points, and for the equation du dt equal f of u. But if with respect to this system of equations, we have two saddle points, and one trajectory departs from one saddle point, another one comes to this saddle point 0, 0. But in general, this is not the same trajectory. So we can change the value of the wave speed c. This is a parameter in this system, and changing the value of the c, we can prove that there exists one and only one the value of the wave speed for which this is the same trajectory. This means that there is a so next result, which is formulated here, the theorem 2, that such in the bistable case, such solutions, reaction-diffusion waves, exist for a single value of speed c, not for many wave speeds as in the monostable case, but only for one speed. And here there are some examples. In population dynamics, rather often we can consider this function f of u written here. And if k equal 1, we have the monostable case shown in the upper figure. And if k equal 2, then we have this bistable case shown in the middle figure. In combustion theory, the nonlinearity is a little bit different, but the same results. So we have very briefly discussed the existence of reaction-diffusion waves. So let us also briefly discuss the existence of pulses. What is pulse? Pulse, by definition, is a stationary solution of this equation with zero limits at infinity. You see at the left figure, it's kind of a bell-shaped function, a solution like that. So since it's a stationary solution of this equation, so this solution satisfies the equation w prime, w second derivative plus f of u equals 0, with zero limits at infinity. As before, we can reduce it to the system of two first-order equations, and we can, as before, study the existence. In this case, it's not a heteroclinic trajectory, it is a homoclinic trajectory, so shown here. Its existence can be easily studied, and we obtain the next result, that solutions like that exist if and only if the integral of the nonlinearity, the integral of f of u, is positive. Well, there are some examples of existence or non-existence of pulses. In particular, pulses exist in the bistable case, not in the monostable case. A very interesting question is related to their stability. So, in fact, such solutions for the single reaction-diffusion equation, such solutions are unstable. For some other models, which we cannot discuss today, such solutions become stable. Of course, what I have presented here, these are very simple results, but the theory of reaction-diffusion waves, of pulses, it's a very well-developed theory. In particular, there are many studies which concern systems of equations and multidimensional equations and local equations and so on, delay equations. And in this case, of course, the questions of existence of solutions, stability of solutions become much more involved and much more interesting, and also many interesting studies of nonlinear dynamics and pattern formation for reaction diffusion equations. So now we begin the second part of this lecture, and we will discuss some biomedical applications, and we will begin with cardiovascular diseases, and more precisely, we will discuss two of them, atherosclerosis and thrombosis. And each time when I will present a new biomedical application, we will start with the physiological background. We need to understand some biological properties of this process. Then we'll introduce a model and discuss some properties of solutions. And then I will briefly discuss some further studies related to numerical simulations or to some other methods of modeling. So let us start with atherosclerosis. Atherosclerosis is a chronic inflammation of blood vessel walls. It can develop in certain areas, in particular certain arteries in our body. In particular, if it develops here in the coronary artery, it is called coronary artery disease, and it happens quite often. So you see what happens here. This inflammation vessel walls become much thicker than they should be because of these fatty deposits. And in the right figure, you see a cross-section of blood vessel walls. In the normal situation, blood vessel walls are very thin. You see this dark gray area here outside. But because of atherosclerosis, the vessel walls become much thicker, and the lumen is area where blood flows. The lumen becomes much more narrow, and because of that, there are less blood coming to, in this case, to the heart, and this can lead to infarction, to arrhythmia, to fibrillation, and to many other complications related to, for example, clot formation and to stroke. And here in this next slide, in the upper right figure, which is how it happens. So I recall that this is atherosclerosis is a chronic inflammation of blood vessel walls. There are very complex processes which are happening here. So what we see here, it's a schematic representation of a cross-section of a blood vessel wall with many cells and many molecules, they call cytokines, involved here, and they lead to the development of this chronic. I cannot stop on this in more detail now. But if we consider this complex biochemical reactions, then we can formulate a mathematical model. And here you see a very simplified mathematical model which consists only of two equations. M, these are cells called monocytes, and A, these are inflammatory cytokines. And we can derive these equations. Then we can study this system of equations. We begin with two ordinary differential equations. And it appears that the very simple analysis, but very important, which shows us that the properties of this model depend on the cholesterol level. If the level of cholesterol is low, then there is a single globally stable equilibrium and the disease does not develop. If the cholesterol level is intermediate, then the system has three stationary points and two of them are stable. This is a bistable case, as we discussed before. So the disease-free stationary point is stable. But if there is initial perturbation which is sufficiently large, then the system can go to another stable stationary point, and in this case, the disease will develop. And the last case, where the cholesterol level is very high, then there are only two stationary points. This is the monostable case, as we discussed before. Disease-free stationary point is always unstable, so necessarily the system will go to this stable stationary point, which corresponds to the disease development. So they begin with this ordinary differential system of equations, but at the next stage we add diffusion response to just mass contain this reaction-diffusion system of two equations, and then in this case we have all this wave propagation as we discussed before in the bistable case, in the monostable case. So in fact what we obtain that atherosclerosis development, this chronic inflammation in blood vessel walls, develops as a reaction-diffusion wave. This is what is important to keep in mind. So summarizing this first analysis of atherosclerosis, you can say that development of atherosclerosis depends on the concentration of LDL, its low-density lipoproteins. Well, it's another name for cholesterol. If for low LDL concentrations, there is a single globally stable point, no disease. And the initial perturbations should be sufficiently large. In this case, the disease will develop. In the last case, if large LDL concentrations, this is monostable wave, so the disease will necessarily develop. And here I show you an example of another modeling of atherosclerosis. This is cellular automata modeling. So what is shown here? So you see this blood vessel, and this red cylinder, it's blood vessel wall, and this green areas here corresponds to the development of atherosclerotic plaques, which narrow the lumen and which decrease blood flow. So this is another cross-section of the blood vessel, and you see here this atherosclerotic plaque is shown as a like white bell-shaped area here, and the blood flow goes above it. And what is important here that this atherosclerotic plaque, it changes the geometry of blood vessel and geometry of blood flow, and this is very important because of several things. In particular, you see there are some vortices here, some flow perturbations, and this is can lead to various to further atherosclerosis progression. But another important point that there is a pressure from the blood flow on this atherosclerotic plaque, and the plaque can rupture. And once it ruptures, we'll see it right now. So it can initiate blood coagulation. We'll discuss it. This model becomes already more difficult. This is what is called fluid plaque interaction, and there are fluid plaque part, solid part of the model, and their coupling. So what I'm saying now, you see is a schematic representation of this atherosclerotic plaque. There's flow here, and because of this flow, the plaque ruptures, and this plaque rupture initiates blood coagulation, and this blood coagulation can lead to arterial thrombosis, and this is what we'll discuss right now. So reduce blood coagulation, and we'll discuss how we can model it. So what is blood coagulation? Of course, it is a very important physiological processes, and if in the case of injury, if the integrity of blood vessel wall is violated, so there is complex biochemical reactions happen here. They lead to formation of fibrin polymer, which covers this area, and also platelet aggregation. Altogether, they protect this injured area and stop bleeding. But as it often happens with physiological processes, if something goes wrong, then there are many diseases related to insufficient or to excessive blood coagulation, in particular deep vein thrombosis and ischemic stroke, and many others. So before starting modeling of blood coagulation, we should explain that there are several components of these processes. First of all, as I mentioned, these are complex biochemical reactions in blood plasma. This is what is called coagulation cascade, and it leads to formation of fibrin polymer, as shown at the top left figure. It's platelet aggregation, and these two processes are related to blood flow. Single reaction-diffusion equations, as we discussed before, for the concentration of thrombin. Thrombin, this is one of the proteins participating in blood coagulation and playing the most important role. And if we study this model as before, we can show that there are these reaction-diffusion waves shown here in the left bottom figure. You see in different moments of time. In the beginning, this thrombin concentration is like that. Then sometime later, it propagates to the right, and so on, so forth. This is a reaction diffusion wave, as we discussed before. And in the bottom right figure, we see numerical simulations in the two domain. In particular, these green half circles correspond to this thrombin concentration in consecutive moments of time. And this coagulation starts in the lower wall because we so as I mentioned, we can describe it by reaction diffusion systems or by one equation in the bistable case, and we have this propagation of reaction diffusion waves which describe clot growth. And then we can compare this modeling with experimental results. For example, we see here in the left figure, this is how this wave, thrombin wave, propagates in the case of hemophilia patient. In fact, this propagation is not sufficient. Either this wave does not propagate at all, or it propagates with a smaller speed. And then if we add some medicals, some treatment, then we can restore normal wave propagation. And this is what is done, in fact, in the clinical situation for patients with hemophilia. So summarizing this, what we just discussed about blood coagulation, as before, blood coagulation is a reaction diffusion wave, and to initiate this wave, we need some initial perturbation which should be sufficiently large. And in fact, this initial perturbation is determined by the pulse solution. You remember we discussed the existence of pulses. So the initial perturbation should be larger than this pulse solution. Then there will be this propagation of reaction diffusion wave, which describes clot growth. Then as a next step of this modeling, we need to consider more physiologically realistic situation, modeling in vivo situation, blood flow. In this case, we introduce Navier-Stokes equations or other equations of motion, and now solving these reaction diffusion equations with these equations of motion, we see here that the form of the clot becomes different. Here in this case, flow comes from the left to the right, so this clot becomes asymmetric because of blood flow. And what is very important is to determine the speed and the final size of clot, and in particular, in the case of excessive clot formation, so this clot can completely occlude the whole vessel and can interrupt blood flow, and this is, of course, is very dangerous. And in this example, well, we introduce also blood cells called platelets, which participate in blood coagulation. So you can see here consecutive stages of clot growth with platelet aggregation. These are these green dots shown here, and this platelet aggregate at this wounded area. After some time, they protect this area, and this chemical reaction can start. You see this blue zone. It's this chemical reaction which leads to the formation of fibrin polymer and so on. So, and here at the bottom, this final clot size, and then we can compare with the experimental results shown at the right figures, but I will not discuss it in detail now. So summarizing about what we discussed about blood coagulation, so these are very important processes, and excessive blood coagulation can lead to arterial thrombosis, in particular in the case of atherosclerosis, to deep vein thrombosis, and to insufficient blood coagulation leads to various bleeding disorders like hemophilia in particular. And there are many important and related questions, in particular chronic inflammation, and what we learned also this is related to coronavirus. Briefly at the end of my lecture. Here I will just only show one example how chronic inflammation influences clot growth, and in particular rheumatoid arthritis, as shown here. Because of this inflammation, there are some procoagulant factors produced because of the inflammation, in particular TNF-alpha or interleukin 17 and some other, which increase blood coagulation. And you see here in the bottom right figure, there's complete vessel occlusion because of this excessive coagulation. And it is known that rheumatoid arthritis increases the risk of cardiovascular diseases by about 10 times. So we stop about blood coagulation, and we'll now briefly discuss in this part of my lecture viral infection and immune response, and in particular viral infection development and spreading, and very briefly about applications to COVID-19. So discussion as before, shortly describe the biological background, in particular about virus replication. Virus, these are small particles which penetrate our body, but they cannot replicate themselves. They need host cells. So this process shown here. So virus penetrates the cell. Its DNA or RNA is copied by the cell, and new virus particles are formed, and they leave the cell to the extracellular medium, and they spread there, and they infect neighboring cells, and like that, viral infection spreads. And in response to viral infection, there is our body. The response of our body is called immune response, which consists of the innate immune response and the adaptive immune response. And here, schematic important stages of this immune response, in particular, this what is called antigen-presenting cells. When they meet these virus particles, they then transmit this information to T lymphocytes and to B lymphocytes, and T lymphocytes become, in particular, this CTL, cytotoxic lymphocytes which kill infected cells, and B lymphocytes produce antibodies which eliminate viral particles. Of course, this is a very brief and very simplified description of the immune response. So we'll begin to study this process with what is called multiplicity of virus assays. These are very standard experiment how viral infection propagate in cell culture, and it is used to study the properties of virus, the response of host cells. So we see this the top view of this petri dish. These white spots occur because of virus spreading here. After some time, infected cells die. This you see here, like this white round spots here. And here it's experimental. So this coronavirus development in the culture of epithelial cells. Epithelial cells, these are cells from our lungs. So in fact, this is a culture of epithelial cells, and we can see how this virus develops here. You see these red areas, it's infected cells. And if we measure the total virus titer, the total virus concentration in cell culture, in time, how it changes in time. So we'll see, like in the right figure, such curves. These are in logarithmic coordinates. So in the beginning, the total virus concentration decreases because the virus needs some time. In particular here, it's about 10 hours to begin to replicate. During this time, it decreases, then it strongly increases, and then it grows, becomes a little bit slower. And if we want to describe these processes, how viral infection spreads in cell, we consider this model. It's a reaction-diffusion system of equations for uninfected cells U, for infected cells I, and for virus V. So what happens here? These uninfected cells U are infected by virus. This is why we have here minus UV, and like that we obtain infected cells, the same term UV with plus. Infected cells can die. This is why we have here this beta minus B and this virus. So it's a reaction diffusion equation for virus concentration. This diffusion term corresponds to random motion of virus. This plus BI, it's how virus is produced by infected cells. This subscript tau here shows time delay. So in fact, it's I of x t minus tau. And virus can also die. This is minus sigma v. So we have this delay reaction diffusion equation for the description of viral infection progression in cell culture. And if we solve the system equation, we can study it by some analytical methods or numerically. And as before, we can show that virus spreads in cell culture as a reaction diffusion wave. This is shown here in the bottom left figure. You see this, if you measure virus concentration in consecutive moments of time, you see this like a bell-shaped function. It propagates from the left to the right. This is how this viral infection spreads in the cell culture. And if we measure the total virus concentration as the integral of this function, the total virus concentration, then we can compare with the experimental result. And this model gives a very good description. For example, in the left figure, you see these red dots. These are experimental results, and blue line, these are numerical simulations. And before, we have 3 stages of the development of viral infection. At the beginning, it decreases, as I explained before, because virus needs some time to begin to replicate. Then you see this very fast burst growth of viral production as the second stage. And then the third stage with a slow growth. This third stage corresponds to propagation of this reaction diffusion wave. So this the next stage of this modeling, we introduce the immune response, and we begin again with this very simple system of 2 ordinary differential equations for virus concentration and for the concentration of immune cell C. So since it is very simple, I will explain what is shown here. So virus concentration is a function of time. So in the right-hand side of this equation, we see here virus reproduction function minus VC. These are immune cells. So this term shows how virus is eliminated by immune cells. And the second equation is for the immune cells. So we have, as before, this logistic term of production of immune cells, but this logistic term is multiplied by a function which depends on the virus. So in fact, it's viral infections. This is why this production of immune cells depends on this function phi of v of depends on virus concentration. And also death of immune cells here can also depend on virus. If virus concentration is sufficiently large, they can kill immune cells. So it's a simple system of two ordinary differential equations, and we can, of course, study it. And the simple analysis shows that there are several stationary points. And then what is very important here, that depending on the parameters and depending on the initial conditions, either we have this trajectory, left trajectory going to low or zero virus concentration. This corresponds to complete or almost complete virus elimination. Or we have this another trajectory going to the right, and this corresponds to chronic disease or some other consequences where there are no or few immune cells. This is called exhaustion. Immune cells are exhausted but also high virus concentration. So this very simple model shows what is most important here. There's a competition between virus and immune cells, and the outcome of this competition, either complete recovery or chronic disease, or even it can be just more dangerous consequences. So this depends on the initial condition. What means initial condition? This is initial viral load. This is very important, initial viral load, individual immune response. So if the individual immune response is strong, disease does not develop or rapidly eliminated. If the individual immune response is weak, so the disease will develop.
Excuse me, you have 5 minutes left. 5 minutes.
Okay, I will finish soon. Okay, then I will go a little bit more rapidly. So these two equations can be reduced to one delay equation, and even this simple equation shows complex dynamics with this transition period doubling bifurcations and transition to chaos. Then if we introduce here in this equation virus motion, we will have this delay reaction diffusion equation and propagation of reaction diffusion waves as shown here. And in particular, there are three different regimes of viral infection spreading. It can be shown by where we have this regime with low viral load, this dotted red line, the regime with high viral load, and the third regime where first we have low viral load and then high viral load. And so, in fact, these are different regimes of propagation. So, as before, viral spreading in cell culture and also in the tissues of human body, these are reaction diffusion waves. And this final example shows how the evolution of viral quasi-species, the emergence of new variants. So the left example shows the evolution, how viral quasi-species can evolve, and here in the right figure the emergence of new variants, viral variants. So to finish, so I have briefly discussed in particular blood coagulation and infection spreading, and these two questions come together when we discuss the coronavirus disease, in particular in the case of lung inflammation because of this coronavirus disease, as shown here. So as we know now, there is excessive blood coagulation and blood clotting in the lung arteries, and this is one of the main mortality causes during coronavirus disease. And in this study, we show how this excessive blood coagulation during coronavirus, shown here, blue line here, it's normal blood, and this red line, plasma from COVID-19 patients. So we see its thrombo formation is much more important in the second case. So to conclude, reaction-diffusion equations is one of the main tools to study biological and biomedical problems. And many physiological processes represent transitions between different equilibria. They are described by reaction-diffusion waves, as we saw in particular for atherosclerosis, for blood coagulation. We have not discussed tumor growth, but this is also the case, infection spreading. And then I'd like to bring your attention to just another type of wave propagation. This is already not reaction-diffusion equations, but it is strongly related to the actual epidemics. In fact, what we see here is it's the data about new daily cases in the world of this COVID-19 disease. So you see here this consecutive waves of this epidemic progression. Now we are at this fifth wave, and what is also important here is that its periodicity about 4 or 5. In this talk, I just recall that there is a very famous painting by Aiwazowski. This is called The Nine Waves, and what is shown here, it's the 9 waves. It's an artistic image widespread in art, journalism, and colloquial speech. Is a symbol of fatal danger, the highest rise of a formidable, irresistible force. So, and in fact, thinking about that, I came up to this kind of poster shown here. So let's not wait. The 9th wave, stop pandemic now. And you see here the 9 waves by Aiwazowski and the 5 wave by COVID-19. And so we should really do massive vaccination to stop this epidemic, to save lives, and to stop the propagation of new variants and these huge economic losses. So I stop on that. Thank you very much for your attention.
Thank you very much. We appreciate very much your expertise that you have shared with us. Agradecemos al doctor Vitaly Volpert por tan interesante plática. Ahora le damos la palabra al doctor Andrés Fraguela para que exprese nuestro agradecimiento al doctor Vitaly y si quiere hacer algún comentario adicional. No lo escuchamos, está apagado su micrófono. Ahora sí.
I would like to say that this is I was waiting a conference like this, very, very interesting, very, ¿cómo se dice? Amplia, no sé cómo se dice amplia.
Broad.
And I have, of course, many questions. I would like to know if the results. I see that you have many interesting results about the viral replications and immune response, and I would like to know if you have also some kind of model to explain the other fast important in COVID-19, the inflammatory phase. You show some one slide about the lungs, the inflammatory process in lungs, but you have also some model for also with the use of reaction-diffusion equations for this phase and the and how it's correlated with the model of the viral replication. This is
right. Yes.
Question.
Okay. Thank you for this question. Yes. So of course, what I showed, it's a just very short, and we could not discuss in detail. And of course, these models were very simplified. Of course, there are more detailed studies which take into account viral replication, more detailed models of immune response, also because immune response, it's extremely complex process. So, for example, if we describe it with ordinary differential equations, there are many equations, but this is very important because with these models, for example, we can describe for example, the duration of incubation period, because this is something which I personally did not understand. And for example, in the beginning of epidemics, there are very different results about the incubation period from several days to several weeks. How it is possible that the same virus gives so different results from the point of view of incubation period? And in fact, with modeling, we can describe that individual parameters of individual immune response for the same virus can give very different incubation period, can give very different and disease progression. So all that, of course, is very important. Mm-hmm. And of course, there are many other aspects of that which I did not discuss. For example, when we study this infection spreading in the lungs, in the epithelial tissue, there is what is called mucus. Mucus is a liquid at the top of the epithelial layer, and it plays a very important role in the functioning of our lungs because it moves, it eliminates like viruses, bacteria, and other different things from our lungs. So it's a complex process, and there is a complex interrelation between infection development and mucus motion. So, and there are, of course, many other aspects. So yes, of course, not only these are very important problems, and we and of course many other people work on them.
Okay. Please, if you can send me your slides, will be very well, because I don't see some titles of the bibliography.
Okay, very good.
You show.
Yes, I will send, of course.
Okay.
I will send you the slides.
And very interesting that you are similar with the picture of Ibasovsky. I don't think about this. It's very interesting.
Yes, in fact, it's just recently preparing the talk. I thought about that, and this is what I really wanted to show. It is, even if it is not reaction-diffusion equations, it's another types of waves, but we should keep it in mind, and we should really make efforts to stop this epidemic. And on our level, this effort is very simple, just to go to make vaccination.
Yeah, very interesting idea. Very interesting. Thank you very much. Very interesting talk.
Thank you.
Thank you very much for enriching our 8th International Conference on Mathematics and its Applications.
Okay, thank you very much again for the invitation, and I wish you a good conference if it continues, and let's stay healthy. Thanks.
Thank you for sharing your expertise with us, and see you next time.
Yes,
okay.
I will be in contact with you.
Okay, thanks. Yes, sure. Bye.